Nikai. I'm co-director for the DC CFAR Basic Science Core, and it's my pleasure to invite you to this annual DC CFAR Basic Science Core conference, which we typically organize once a year. So the goal of our Basic Science Core is to support collaborative HIV research by basically providing various core services and facilitating HIV research and involving more and more investigators into this research. So I'll talk about our services at the very end of this meeting. So please stay put. So our CFAR is unique because it's city-wide CFAR. So it includes essentially all Washington D.C. Scientific institutions. So this includes George Washington University, Georgetown, Harvard University, Children's Hospital, American University, Veterans Affairs, Whitman Walker Clinic, and DC Health. I hope I didn't forget anyone. So this meeting will be structured that you'll listen to our keynote presenter, who this year is Dr. Mohamed Abdel Mahsen. He's associate professor of vaccine and immunotherapy at Wistar Institute in Philadelphia. And this will be followed by the pilot award recipient, Dr. Campbell, and also by the T32 trainees by Mr. Griffin Greco and by Mrs. Teresa Murphy. So at the end, as I said, I will highlight our core services and talk a little bit about our finding opportunities. And basically, I would like to thank you for your interest. I hope that this meeting will be helpful in terms of facilitating collaborations, writing more papers, submitting more grants. And before we go forward, I also would like to acknowledge my co-directors. I would like to mention Dr. Marta Catalfama from Georgetown University and Dr. Alberto Boschi and Sanjay Magiwar from the George Washington University, who are co-directors of this Basic Science Core. I also would like to acknowledge our core manager, Vivian Kirkham, who helped us to organize this meeting, and also our immediate previous manager, Cara Thompson, who was helpful also to facilitate this meeting. And I would like to also to thank Pete Simon, who just left left CFAR, who was very instrumental in setting up this meeting, and current admin director, Brandy Robinson, for the help in organization. So before we start, I'd like to introduce our speaker, who is Dr. Abdel Mahsen. He received his bachelor degree in microbiology and then master's degree in virology in Cairo, Egypt, at Ain Shams University. And then he moved to UCSF, San Francisco, where he was duly supervised by UCSF and also by Ain Shams, and he received his PhD in virology. So he did 2 years of postdocs at the Blood System Research Institute in San Francisco. And then in 2017, he joined Wistar Institute as a junior faculty. So basically, he investigates the role of glycans in regulating immune response and inflammatory response during the HIV infection. He rose through the ranks, and now he serves as a co-director of the virus and reservoir core lab. And he also plays a key role in the overall CFAR strategic planning at Wistar and the University of Pennsylvania. He was promoted to the associate professor in 2022. He received many awards, including CFAR Early Career Award of Excellence in Basic Science, and also he received 4 consecutive Young Scientist Awards at CROI. He has been speaker and plenary lecture presenter at many national and international conferences. So he did several significant contributions to the HIV research, including developing ultrasensitive PCR and deep sequencing techniques for the analysis of the drug-resistant mutations. He also investigated the role of host factors, including P21. This led him to develop his current research, which is basically looking at the glycosylation and its role in HIV infection. And he also actively collaborated through the PEN CFAR and also participated in the Martin Delaney collaboratory. So he published over 70 papers, and he is supported by 201 grants. And I think with all this said, I'd like to invite Dr. Abdel Mahsen to present his keynote lecture. Thank you.
Thank you very much, Sergey, for the introduction, and thank you for inviting me to give this presentation. Just let me know if you can hear me well and see my presentation in full screen.
I think it looks good, yeah.
All right, great. So yeah, I'm very excited to have the chance to share with you some of the work we are doing in our lab. And in our lab at the Western Institute, we work in the interface between the fields of virology, immunology, and glycobiology. And it's pretty obvious why we need to understand immunological functions during viral infection, but what is glycobiology and why should you care? So I would like to start with a very simple fact that there is not a single living cell on Earth that is not covered with a heavy, dense layer of sugars, glycans on its cell surface. And some of these glycans or sugar get secreted or shed into the circulation. And in the last few years, it became clear that these glycans, both cell surface and cell-free, are biologically active molecule that can play an important role in modulating several cellular function and immunological responses that are central to many diseases, including cancer and infectious diseases. And there is a lot of fascinating work being done in this area within the cancer research, but not as much in the infectious diseases field, including virology, including working in HIV. We all know, of course, that is the glycan shield, the sugar that has covered the HIV. The virus itself has been heavily studied for the last maybe 30 years or more, mainly for vaccine developing purposes. But on the other side, the role that the host glycans may play in regulating immunological responses that are important for HIV resistance and immunopathogenesis has been heavily ignored, despite its potential for therapeutic applications. And that's why within the work we are doing in our lab that focuses on HIV, because we work in other viruses as well, it's centered around this question, how host glycans can impact immunological and inflammatory function during our suppressed HIV infection. And this work can be divided into two interconnected areas of research. One focuses on how these host glycans can impact inflammation and aging-associated comorbidities that we all know is still prevalent in HIV-infected individuals despite long-term therapy. And in fact, actually, most of the work we are doing in our lab falls under this area of research because these glycans are a very potent modulator for inflammation and biological aging. However, for the time today, I will skip this part and I will focus on the second area, which is how these glycans can impact viral resistance. And today I want to give you two quick examples. One focuses on cell surface glycans and the other focuses on circulating glycans. And within each one of those two examples, I want to show you how we can start with a glycomic approach to zoom into a biological function that we think important for HIV persistence. So I will start with the first example on cell surface glycans, and in particular, how these cell surface glycans could be modulators of antiviral innate immune functions, which is some of this work was published some months ago in this paper in PLOS Pathogen, and it was led by Samson Adeniji, a postdoc in our lab. So I'm sure you're all familiar with immune negative checkpoints, classic immune negative checkpoint that can impact immune function by binding to a receptor on the surface of cancer cells. For an example, PD-1 expressed by immune cells bind to PD-L1 on cancer cells, and this binding can inhibit the function of immune cells and allow cancer cells to evade immune surveillance. However, recently it became clear there is another class of immune checkpoints that exert their immune suppressive effect by binding to sugars, to glycans on the surface of target cells, and that's why they call glycoimmune checkpoints. So again, let me start by telling you that is every single immune cell express on their surface several sugar binding proteins, or also called lectins. And some of these lectins play a very important role in the ability of these immune cells to identify and target their target cells. So the binding between these lectin on immune cells to the sugars on the surface of target cells play a very important role in the ability of these immune cells to identify and target these cancer cells or virally infected cells. And that's why several cancer cells or virally infected cells in purpose decorate themselves with certain sugar, which is the ligand for this glycoimmune checkpoints, in order to ensure evading immune surveillance and hiding from the immune system. And that's why there is a lot of interest in target these glycoimmune checkpoints to enhance immune surveillance and targeting cancer cells. So this work started in our lab a couple of years ago when we did a full glycomic analysis on the cell surface of HIV-infected cells, with the hypothesis that is the cell surface of HIV-infected cells during therapy will have a unique glycomic signature to it. And taking a long story short, because this work was published a couple of years now, we found that is the cell surface of HIV-infected transcriptionally active cells during therapy is indeed have a glycomic signature to it compared to the cell surface of HIV uninfected cells and HIV-infected transcriptionally inactive cells. And one of the features of this glycomic signature is an upregulation of this sugar called alpha-2,3-sialic acid. And that was interesting for us because this sugar, alpha-2,3-sialic acid, is the ligand of one of these glycoimmune checkpoints called cyclic 9, which is a sugar binding protein bind to this particular kind of sialic acid. So cyclic 9 protein is expressed on the surface of many immune cells, including in a subset of the CD56-DIM cytolytic NK cell. And the binding between cyclic 9 and NK cells to alpha-2,3-sialic acid on target cells should inhibit the ability of these NK cells to target these target cells. So now knowing that his HIV-infected transcriptionally active cells have a high level of the ligand of cyclic 9, we wondered if this is a way for HIV-infected cells to evade immune surveillance. So we asked the question, do cyclic 9 sialic acid interaction contribute to the ability of these HIV-infected cells to evade NK immune surveillance? And then we asked, if we do find that this interaction contribute, can we design a selective approach to break this interaction and enhance the susceptibility of HIV-infected cells to NK-mediated clearance? But before we even start answering these two questions, we wanted to do a simple test. We reasoned that if sialic acid cyclic 9 interaction plays any role whatsoever in HIV infection, we should see a correlation in vivo in human between the frequency of CD56-DIM NK cells that express this inhibitory receptor cyclic 9 here in the x-axis in both panel, and the level of HIV in the y-axis, either level of HIV RNA, plasma viral load when the patient are viremic, or the level of cell-associated HIV DNA measured in isolated CD4 T cells when the patient are suppressed. And we reason that is, we should see a positive correlation between the level of this NK cell that express an inhibitory receptor and the level of HIV. You know, more inhibition, more HIV. But to our surprise, in both cases, we see a significant negative correlation, which didn't make any sense to us. Why more inhibition or more inhibitory NK cells will lead to less HIV? And again, that doesn't make any sense to us. And at this point, we decided to ignore these correlations as maybe just a random in vivo correlation, which happens. And until this paper came out where the author investigated the role of the same NK subpopulation that expressed cyclic 9, but now during in the context of hepatitis B viral infection. And in this paper, the author showed this correlation, a negative correlation between the level of the same NK subpopulation that expressed cyclic 9 and the level of hepatitis B viral DNA. Unfortunately, the author didn't try to explain this apparently doesn't make sense correlation, but it was striking for us in how similar it is to the correlation we found with HIV, and made us wonder if this is actually a true correlation that does have a biological significance, but we just don't know what it is. And it was at this moment that is, it came to us maybe that we are confusing the subpopulation with the molecule. Now, the molecule, which is cyclic 9, is inhibitory, but that doesn't necessarily mean that it's the NK cell or subpopulation that express this molecule are also inhibited, because the cytotoxicity of NK cells is a balance between the expression of several activating markers and receptor and several inhibitory marker and receptor. And it may be possible that is this population, despite being expressing the inhibitory receptor cyclic 9, do express high level of other activating markers, maybe low level of other inhibitory marker, and maybe these cells are actually more cytotoxic against HIV than the Siglec-9 negative cells, which would explain this negative correlation. And if you really think about it, that is not very weird, because usually immune checkpoints like PD-1 are expressed on highly activated, highly differentiated T cell, and they expressed on it in a way to restrain their activity. So we start exploring this alternative hypothesis, that is, these cells, despite being expressing inhibitory receptor, are actually activated and cytotoxic against HIV-infected cells. And we start exploring that by measuring the expression of several NK activating receptors and markers on the surface of Siglec-9 positive cells here in solid circle, or Siglec-9 negative cells here in open circle. And those NK cells were isolated from HIV-infected and uninfected individual, HIV-infected or suppressed individuals, on HIV-infected viremic individuals. And as you can see here, we measure the expression of CD16, which is very important, actually the receptor responsible for mediating antibody-dependent cytotoxicity, CD38, NKB30, peripherin, and DNAM, all activating markers and receptor. And in each one of those cases, indeed, the Siglec-9 positive cells expressing higher level of these activating markers and receptors compared to the Siglec-9 negative cells. We also looked at inhibitory receptor, NKG2A, which is a very potent inhibitory receptor for NK cells, and TIGIT. And again, in both cases, the Siglec-9 positive cells express lower level of these inhibitory receptor. So what we know so far that is these cells, despite being expressing Siglec-9, have an activating phenotype in vivo, and they do correlate negatively with HIV. Again, both things supporting the fact that the idea that is these cells are more cytotoxic against HIV compared to Siglec-9 negative cells, something we wanted to explore. So we sorted Siglec-9 negative cells, Siglec-9 positive cells, and we co-culture them with a cell line called CEM-NKR, which is naturally resistant to HIV, to NK killing, until you infect them with HIV. And you can see here that is indeed the Siglec-9 positive cells showed higher cytotoxicity toward HIV-infected cells compared to Siglec-9 negative cells. They also showing higher degranulation as measured by CD107A expression. And this observation has been repeated multiple times, including the use of autologous assay and primary NK cells with autologous primary CD4 T cell. So we know that is the Siglec-9 positive cells are more activated, have more activated phenotype in vivo, are more cytotoxic in vitro than the Siglec-9 negative cells, which is now consistent with our negative correlation with HIV RNA and DNA in vivo. But the next question is, what about their expression of Siglec-9? Now, these cells are cytotoxic, but they still expressing an inhibitory receptor. Is that possible that is this inhibitory receptor is still restraining the otherwise will be even higher cytotoxicity of these Siglec-9 positive cells? These cells have other feature that is allows them to be cytotoxic, but now they have this kind of break that is making, that doesn't allow them to take a full potential of their cytotoxicity. Is that possible? So what we did is we developed a blocking antibody for Siglec-9, and we co-culture NK cells, total NK cells with HIV-infected cells in the presence of either an isotype control or this blocking antibody. And you can see definitely blocking Siglec-9 interaction dramatically increasing the already high cytotoxicity of the Siglec-9 positive cells against HIV-infected cells. And this was Siglec-9 dependent, because if we use Siglec-9 depleted NK cells as an effector cells instead of the total NK cells, this effect, we don't observe this effect anymore. And again, this was against a cell line infected with HIV, but we could recapitulate the same data using an autologous assay where we isolate CD4 T cells from PBMCs of healthy individuals, infect these CD4 T cells with HIV, and then co-culture them with NK cells from the same individual in the presence or absence of the Siglec-9 blocking antibody or isotype control. And as you can see here, looking at the NK degranulation, indeed the Siglec-9 positive, Siglec-9 blocking antibody enhances inducing the degranulation of NK cells against autologous primary CD4 T cells, and mainly it's increasing the degranulation of the Siglec-9 positive cells, not the Siglec-9 negative cells. This is also combined with a reduction in the level of P24 positive CD4 T cells in the co-culture. And when we take the supernatant from this co-culture and try to infect TZMBL cells, you can see that as a supernatant from the co-culture that happened in the presence of the cyclic-9 blocking antibody in red, having lower infectivity compared to the co-culture in the presence of the isotype control here in the gray. So what we know from this work that is surprisingly, the cyclic-9 positive cells are actually having a highly activated phenotype in vivo, and they are highly cytotoxic against HIV-infected cells compared to the cyclic-9 negative cells. And that is maybe because they are expressing high level of several NK activating markers and receptor, including NKB30, DNAM, compared to the cyclic-9 negative cells, and maybe expressing low level of NKG2A, which is inhibitory receptor, again, compared to cyclic-9 negative cells. However, this high cytotoxicity of the cyclic-9 positive cells still being restrained by the cyclic-9 molecule and its inhibitory effect on in the NK functionality. So that leads me to the next question. Can we develop an approach that selectively target this interaction? Obviously, you might think that is we can use a blocking antibody for cyclic-9 we developed. However, this cyclic-9 expressed in other immune cells, including myeloid cells, and blocking the cyclic-9 interaction in these cells randomly might cause inflammation. So we wanted to develop an approach that is selectively target these interactions for HIV-infected cells alone. And we thought that is maybe we can use an enzyme called sialidase, which remove sialic acid to remove the ligand of cyclic-9 that overly expressed on the surface of HIV-infected cells. But again, the sialidase will go and remove sialic acid from all cells, which can cause unwanted side effect. So we reasoned that is we can conjugate this enzyme, the sialidase, to HIV specific antibody. In particular, we use a broadly neutralizing antibody. And we reason that is this BNAb, this broadly neutralizing antibody, will steer the sialidase to the surface of HIV-infected cells that is hopefully expressing viral antigen after an efficient shock and kill or during our cessation. And then the sialidase will remove the ligand for cyclic-9 from the surface of HIV-infected cells without impacting HIV-negative cells, which could potentiate NK killing of these target cells. So indeed, we went ahead and we did several versions of this BNAb sialidase conjugate using antibody like 3BNC117, 101074, and NIH 4546. And then we first ask us a question, does this conjugate, can this conjugate remove sialic acid selectively from the surface of HIV-infected cells without impacting HIV-negative cells? So what we did, we mixed HIV positive cells here in right and HIV-negative cells here in the left in the same tube, and then we treat them with the antibody alone. So you can see this both bibulation, and we stain them for sialic acid in the y-axis, and then we treat them with a low concentration of the BNAb sialidase conjugate, and you can see clearly that is the sialic acid from the surface of HIV-infected cells are being downregulated without impacting the sialic acid from HIV uninfected cells. We can increase the concentration of the conjugate, and we can see a nice dose-dependent reduction of the level of sialic acid from the surface of HIV-infected cells with minimal impact on HIV uninfected cells. So that's good, but whether this removal of sialic acid from the surface of HIV-infected cells will potentiate NK killing against these cells is what we explored next. And how we explore that, we first use the cell line, the HIV uninfected cells or HIV-infected cells, and we stain them or dye them with the HIV uninfected cells with a red dye, HIV-infected cells with a green dye, and we mix them and we use live imaging staining, and we can see the number of the cells as well here. So this is a culture of HIV-infected and uninfected cells. We can add NK cells from HIV-infected or suppressed individuals, and you can see there is a reduction, about twofold reduction in the level of HIV-infected cells without impacting HIV uninfected cells. So that's like the cytotoxicity of these NK cells. We can add an isotype control and nothing happened. It's still twofold, remain twofold. We can add two different concentration of the BNAb at 200 nanomole or 400 nanomole, and you can see this twofold become 5- or 6-fold as a result of antibody-mediated cytotoxicity of this BNAb. And you can clearly see the number of green cells are dramatically reduced by this BNAb. So do we even have a room for the BNAb sialidase conjugate to even reduce the green cells even more. Indeed, we do actually now if we use the exact same concentration of the BNAb, but now with the BNAb sialidase conjugate, you can see the 5-6-fold become about 50-fold reduction, and you barely see any HIV-infected cells in this co-culture. But even more importantly, this happened with a minimal impact on HIV-uninfected cells. Here in red, you can see them is still the same. So that's actually good. And this is using a cell line. We wanted to repeat that again in autologous primary cell assay, where we use effector cells and target cells from the same individuals, and we examine either NK degranulation, either as the expression of CD107 co-expression with interferon gamma or TNF alpha. And you can see here the BNAb sialidase conjugate at 2 concentration here in bright red elicit higher NK degranulation compared to the same concentration of the BNAb here in dark red in all of the CD107 expression or co-expression with TNF alpha or interferon gamma, and that was reflected in a lower level of HIV-infected cells measured by intracellular B24 expression in these conjugate. So this is just one example, again, in how we can start with a global glycomic analysis on the cell surface in order to identify a new immunological function that cyclic 9 sialic acid axis could be a glycoimmune checkpoint mechanism that can be exploited by HIV-infected cells to evade immune surveillance, and how this information could be applicable and useful, and we could use them to develop a proof of concept approach to selectively enhance the susceptibility of HIV-infected cells to immune-mediated clearance. And obviously, we are trying to explore how this conjugate will behave in vivo in animal models as we speak. But that is actually conclude the first example on cell surface glycans and lead me to the second example in circulating glycans. And I showed you how we do a lot of work trying to identify a cell surface if you would say, biomarker of HIV resistance with a potential functional significance that is we can exploit. And we do the same thing with cell surface, with circulating glycans. Again, how these cell-free glycans could be used as a functional biomarker of viral resistance, which is, again, the work that was published last year and was led by Leila Giron, a scientist in our lab. But before I show you this work, I just want to spend a few seconds telling you why we even need to identify a cell-free biomarker of HIV persistence. So, of course, all of you know that is upon treatment interruption, ART interruption, in vast majority of people, the virus rebound very quickly within a couple of weeks. However, recently, it become clear that there is a rare population of individuals, termed post-treatment controllers, who can maintain viral remission for several months or years after stopping therapy. However, we don't know how they do what they do. And recently, there is a lot of interest in identifying biomarkers that can be measured before stopping therapy and can predict either time to viral rebound or probability of viral rebound, because in one hand, these biomarkers can be used to mitigate some of the risk associated with analytical treatment interruption, but even more importantly, they can give us some biological clues into the host mechanism contributing to remission. And we have obviously focused our attention into exploring this for exploring cell surface glycans and metabolites. And the reason why we thought that is cell-free glycans and metabolite. And we thought those cell-free glycans and metabolite could be helping us achieving this goal, because these glycans and metabolite comes to the circulation, come to the plasma by either active secretion or leakage from multiple tissues, organs in the body. And research in other diseases have shown that their levels and chemical structure can reflect the overall status of these tissues that they are coming from. And obviously we know that HIV is an inflammatory disease. So they can be used as a good biomarker of tissue inflammation. But on the other hand, as I mentioned, these glycans and metabolite are biologically active molecules that can be taken by the receptor in the surface of immune cells, those lectins, to start a signal transduction that can impact the fate of these cells. So in one hand, they could be a biomarker of inflammation, but on the other hand, they could be a driver and mediator of both inflammation and immunological function, making them an excellent candidate for a functional biomarker discovery. So taking a long story short, we used samples from 3 independent cohorts of analytical treatment interruption that had samples from 8 different clinical trials in the US and South Africa. And using these samples, we could indeed identify and partially validate a set of cell surface glycans and biomarker and metabolite that their level before stopping therapy associate with either a longer time to viral rebound or a shorter time to viral rebound, even after adjusting for key demographic and clinical confounder. And I'm not going through this work in details for the time, but I want to highlight 3 things about these potential biomarker quickly for you. The first is that they were not randomly distributed. At least the metabolic biomarker were not, and they were belonging to a specific metabolic pathway. The metabolite that is predicted at the delayed rebound were all embedded into anti-inflammatory metabolic pathways, including glutamate metabolism and whatnot. And the marker that is predicted accelerated viral rebound were all embedded into pro-inflammatory metabolic pathway, including those that is associated with gut microbial dysbiosis and translocation. Just to give you a couple of examples of those, this metabolite, a pro-inflammatory metabolite called TMAO, is established pro-inflammatory metabolite, and its level was higher in the people delayed fast compared to the people delayed later, and this is a metabolite related to the gut microbiome dysbiosis. Another is multiple metabolite that part of the tryptophan metabolism pathway, and the tryptophan catabolism or metabolism pathway is not only a marker of gut microbial dysbiosis during HIV infection, but it has been dysregulation in this metabolic pathway has been associated with the development or the maintenance of several aging and inflammation associated diseases during HIV, including subclinical atherosclerosis, diabetes, liver fibrosis, and whatnot. And I think this is interesting because it's one of the very few evidence that there might be a shared host pathway between the development of HIV-associated comorbidities and HIV persistence, or at least time to viral rebound. The second thing I want to highlight about this potential biomarker is that we were lucky to have access to large number of post-treatment controllers. Those are very rare. I think there is only like less than 100 of them identified in North America. And we had an access to about 27 of those that were selected by Jonathan Lee at Harvard. And that allowed us to ask the question, do the level of some of these marker can differentiate who will become post-treatment controller PTC from non-controller NC. And as you can see here, indeed, the pre-ATI level of several of these markers are significantly different between post-treatment controller from non-controller. So next, we wanted to use a machine learning algorithm to select from these markers a smaller set of markers that can be combined together in a multivariable model. And the machine learning algorithm selected those 7 markers that is, when combined together, could differentiate who will become post-treatment controller from non-controller with about 97.5%. We were worried that is overfitted, so we tried that with several other modeling system. And in any case we tried it to do it, it doesn't go down to below 94.7%. And this is translated in a score, and you can see here this score can differentiate who will become non-controller from post-treatment controller with a high sensitivity, specificity, and accuracy. So we are following up on this potential marker, but from this work, we identified a potential pre-treatment interruption non-invasive plasma biomarker that may predict duration and probability of viral remission after treatment interruption, even after adjusting for key demographic and clinical confound. The last thing I want to highlight for you about this potential biomarker before I wrap up is that, as I mentioned to you, one of our main interests in looking at plasma glycans and metabolite, that is the biologically active molecule, increasing the chances that is they do have a function in the phenotype we are observing. And indeed, several of these glycans and metabolite that is ended to be in the multivariable logistic model, we tested in vitro, and they do have an impact on HIV. I just want to give you one quick example, which is this metabolite called L-glutamic acid, which we found in vivo to predict a delayed rebound and was higher in post-treatment control arm compared to non-control arm. So we asked the question, is it possible that this L-glutamic acid is actually functionally contribute to the phenotype and maybe impacting HIV transcription? So we start by exploring its impact on HIV, latent HIV transcription using the GLAT model, which is I'm sure many of you are familiar with, but for whom of you are not, it's just a Jurkat cells having HIV integrated into their genome with GFP under the control of LTR. So when you reactivate them with B-aminomycin or TNF-alpha or anti-CD3, anti-CD28 and some clones, they just become green, and you can look at that in the flow cytometry. So what we did is we stimulated these cells in the presence or absence of L-glutamic acid using glutamine-free media. And indeed, as you can see here, the B-aminomycin here in black reactivates those cells very well. Same thing with anti-CD3, anti-CD28. But the addition of L-glutamic acid dramatically inhibit the ability of these stimulus to reactivate latent infection. Obviously, this was done in the GLAT cells, which is very useful tool, but lies a lot, and we needed to confirm that in primary cell. So we isolated CD4 T cells from HIV-infected R-suppressed individual, stimulate them with B-aminomycin, which is the most potent way to reactivate these cells, again in the presence or absence of L-glutamic acid, and measure cell-associated HIV RNA as a marker of latent HIV reactivation. And again, consistent with the in vitro data in the GLAT, the addition of L-glutamic acid here in orange inhibit or partially inhibit the ability of B-aminomycin to reactivate latent infection, which is interesting because now we could, by doing a full omic analysis, zoom into a marker that's have a functional, a direct mechanistic functional impact on HIV. And we then ask us the question, how L-glutamic acid might be doing that to HIV transcription, inhibit HIV transcription. And we know that is inside the cells, L-glutamic acid, or also called glutamate, enter the TCA cycle by being converted to another very important metabolite called alpha-ketoglutarate acid. So we wonder if alpha-ketoglutarate acid plays a role in the phenotype. And what supported this hypothesis is that when we went back to our patient data, we indeed found that is the individual who have high level of alpha-ketoglutarate acid tend to rebound later compared to the individual have low level of it. And indeed, when we use different concentration of alpha-ketoglutarate acid in the GLAT system, you can see a nice dose-dependent reduction in the ability of B-aminomycin to reactivate latent infection. Then we know that is glutamate convert to alpha-ketoglutarate acid through an enzyme that can be inhibited using this inhibitor called EGCG. So we try to see if we use EGCG, can we inhibit some of the L-glutamic acid mediated inhibition of HIV transcription, which would suggest that is L-glutamic acid work by being converted to alpha-keto. And indeed, as you can see here, when we add L-glutamic acid to B-aminomycin, that inhibit the ability of B-aminomycin to reactivate, which is quite consistent data. But when you add, when you do that in the presence of EGCG, some of this inhibition is being reversed, suggesting that it's at least partially L-glutamic acid working by being converted to alpha-ketoglutarate acid, which is actually quite interesting because alpha-ketoglutarate acid is known as a master regulator of the epigenetic landscape of immune cells, including CD4 T cells. And we know that is, of course, that is HIV transcription is being mediated by epigenetic landscape of the cells, which is something we're currently investigating to see if the impact of alpha-glutaric acid and epigenetic modulation of the cells is behind its impact on latent HIV. But again, this is just another example of how we can start with an omic approach to zoom in on a cellular function that we think is important for HIV. And with that, I will just close that hopefully I could convince you that is glycans are important. And if you are interested more in this topic, I will refer you to this review article we wrote a couple of years ago, where we actually discuss 4 different areas of investigation that we think the HIV field can take advantage of the recent advances in the glycobiology field. And I will refer you also to the different publication we had in each one of those different areas. And with that, I will go to my acknowledgement slide. I first and foremost thanking all of the volunteer participants in our different studies. I will thank folks in our lab. I had the chance today to show you work from Leila and Samson, but hopefully next time I can show you work from Britt, Majin, Angela, Shalini, and Ferlina. I would like to thank our collaborators, with and outside Wister, our funders, and we'll be happy to take any question, and also feel free to email me with any potential collaboration, idea, thoughts, even random thoughts. I will be happy, and thank you.
Great, thank you, Mohammed. Thank you for a very nice presentation. So we can take a few questions if anyone has a question. I can start. So I think my question was, we've been looking at the HIV infection through, like, we hypothesized that HIV envelope has glycans which can interact with the glycans on the cell surface, and this process may be important for viral infection. So have you looked at that, or have you?
Yeah, we actually just published a paper together with Nadia Ruwan in Gladstone Institute, utilizing a technology that we developed together called cytoph-leak, which is doing cytoph, but instead of looking only at the protein, we look at the protein and the glycans. In this work, we definitely found that is certain glycans on the surface of CD4 T cells are very important for HIV infection. And that was actually done within the memory CD4 T cells, because we know that the memory CD4 T cells are infected at higher level than naive. But even within the memory CD4 T cells, there are certain glycans that is increasing the susceptibility of HIV infected cells to infection. We are now trying to understand how that might be linking to persistence, but the infection itself is definitely we know the virus itself have its own glycans, but also the interaction between the virus and the host cell surface glycans on the surface of CD4 T cells are indeed playing an important role, that when we just remove one single sugar from the surface of this CD4 T cells, we see a multi-fold reduction in the infectivity of these cells. And again, this is all within memory CD4 T cells, not just like, it's not just a marker of naive versus memory. So I think that is very interesting, because there's very few proteins that are known that is also causing this different susceptibility. So the fact that there's one sugar can do that is very interesting. And as I mentioned, this is just a paper we published maybe just like 2-3 weeks ago in eLife, but we just used that for active infection, and we currently doing the same thing for latent infection and whatnot.
Thank you. Yeah, very nice. Yeah, I'll take a look. Maybe we can at some point discuss. Yeah,
absolutely.
Great. Yeah. Anyone else, please? Yeah, and especially trainees. Yeah, please ask questions. Yeah.
Sorry, I have a quick question. Wonderful presentation. It was very interesting. Thank you for being here. So I have a quick question regarding the first part of your talk, the siglet. You mentioned that that infected cells express this specific glycosylation in this receptor. I was wondering if you look in the difference of the immune activated cells, for instance, if this molecule that potentially is expressed shows the same type of glycosylation, or you expect to see, that is totally HIV specific?
I think that's an excellent question. And in the cell report paper, we have investigated this in details, because obviously in order for the cells to be infected, it needs to be activated, and T cell activation causing so many different changes to the cells, including glycomic changes. So indeed, T cell activation changing the glycans, but the HIV infection by itself does do it. So we actually explore that using two different ways. We infect CD4 T cells without activating them, and we use microbeads with CD44 to enhance the infection, because it's so hard to infect CD4 T cells unless you activate them. But we spend some time to look at different protocol that would allow you to have high level of infection of CD4 T cells without activating them. And we have several figures in the paper showing that is HIV infection, T cell activation does not induce that particular sugar, and that HIV infection in resting CD4 T cells does induce it. So it's HIV specific. But we, in an unpublished data, we took it even one step further. We know that is when we infect HIV with CD4 T cells with NL4-3, these glycans go up. So, but how? We don't know. So we actually did make a plasmid with every single HIV genes, and we transfect CD4 T cells with each one of them, and we now know that it's only VBR can induce this sugar. So it's not T cell activated, and we even know that is one single accessory genes of HIV or regulatory gene that's enough to induce the sugar. Why and how? Obviously, that is not a known function of VBR, but it might be an additional function that is we didn't know about before, something to allow these infected cells to evade immune surveillance. So it's an HIV-specific signature. However, T cell activation have its own signature that is separately.
Mm-hmm. And the last question, just as a curiosity, because, you know, we live in the era of COVID. So does COVID induce this type of immune activation? You see have you seen this type of difference in this particular molecule in other viral infections?
Absolutely. We actually have a paper under, it's not under review yet, still with editor, to show that is almost identical story to what I showed you with HIV is happening with hospitalized COVID patient. They also have the same increase in sugar. Cyclic 9 allow the SARS-CoV-2 infected cells to evade NK immune surveillance, and we can increase the NK capacity to target those SARS-CoV-2 infected cells by blocking cyclic 9. So it is not, and I showed you obviously it's happening also with hepatitis B. So I don't think it's an HIV specific mechanism. Cancer use it, and apparently different viral infection also use the glycoimmune checkpoints to evade immune surveillance.
Okay. So thank you so much. Wonderful work.
Thanks.
Any trainee has any question?
Yeah.
I'm pushing that again.
Please, yeah.
Hello. Yeah, thank you for that presentation. About your antibody constructs against the salicylic acid, did you just conjugate the enzyme directly to your antibodies of choice, or how did you do it?
Yeah, we collaborate with a bioengineer at Rice University. His name is Zeo Han, and he developed this technology called B-Click, which allows you to conjugate anything to the antibody without changing the antibody. So you don't impact the antibody capacity to, elicit their neutralizing and non-neutralizing function. So it was, directly conjugated to the antibody using this B-Click technology, and that was detailed in the methods of this paper.
Okay, and also, if it's privileged information, you are free not to answer or to say it's privileged information, but we all know that there are various BNAbs and they all have their different efficacies, so to speak. So which ones did you have most success with? We
use 3BNC117. In the paper, we use 3BNC117, BGT151, and NIH 4546, and all of them were quite comparable to each other. However, we continue working with them now in animal model, and they are having different PK and efficacy in the animal model, but it's too early for me to know which one is the best in vivo yet. But we are moving forward with all of those three, and we actually, not three only, we actually added 1074 to the mix, because 3BNC117 and 1074 has been currently heavily used in the clinic. So we wanted to test that as well. All of them work quite well in vitro, but which one of them will be better in vivo, that is to be seen hopefully soon.
Okay, and final question, sorry. As you already said, the virus also expresses glycans. And you're targeting actively producing virus producing cells. So do you think that the viruses that are battened out could in a way capture the antibodies that you are using to treat the cells, that not enough will get to the cells? I don't know if you understand what I mean.
But the idea of actually with the antibody is we want it to go to the virus that is budding out from the cells. We want that to happen because that is the only way for adding selectivity to the cellulase. We want the antibody to definitely go to this viral budding. Oh, maybe I misunderstand. You mean that is a virus that's already came out from the cell?
Yes, that's what I mean.
I do think that is maybe that's possible. That's why we will need to try different concentration of the antibody to make sure. However, you know, with patients suppressed on antiretroviral therapy, if they are not stopping antiretroviral therapy, the number of infected cells and viral production is not that high to, I think, occupy all of the BNAB you will give it to them. But even if that happens, it probably, if I think correctly, it will be a matter of changing the concentration to make sure that is you, yes, cover the virus that is floating, but there is still enough BNAB cellulase conjugate to go to the one that is coming out from the cell. Yeah.
Okay. Thank you very much.
That's to be tested.
Any other question?
I have a question. So, so these sialic acid are kind of, are very profoundly found on the cell adhesion molecules, right?
Yeah.
And as I understand, they do change over like age as you age. So does that hamper or affect, or does that point needs any attention while you?
Yeah, you are absolutely right. So glycans are very active as in aging. And as I mentioned, most of the work we are doing in our lab is actually focusing on aging, but I had to skip that. But definitely we found that is HIV, as people getting older, they lose different anti-inflammatory glycans and they gain many pro-inflammatory glycans, including losing sialic acid. But that's in the general. We do find during HIV infection that is, they do have lower level of sialic acid, but not on the surface of HIV infected cells. I'm not sure why is that, but in general, if we look at in the HIV infected individual in their IgG, plasma glycoprotein, the gut, and we have published in all of this, we found that they lose sialic acid, but not in the HIV infected cells itself when it's transcribing the virus, because apparently the virus itself increasing sialic acid in these few cells. But obviously how the loss of the sialic acid, which is pro-inflammatory in its own merit, impacting aging and inflammation is a very active area that is we are investigating as we speak. But the HIV itself, the HIV infected cells itself doesn't have this phenomena, which is might be why they can, not why, but like part of why they can evade immune surveillance. But aging definitely impacted glycans, and actually HIV plus aging impacted even faster. It's increasing the base of losing this anti-inflammatory glycans.
Okay, thank you.
Maybe one more question, right? And then I think we have to move forward.
Yep.
If no trainee will take it, I will take a last question if it's possible. On the delay in rebound that is and the metabolites that are associated, anti-inflammatory metabolites associated with that, do you think that there is any role of diet or the microbiome and things like that? It is that the those individuals that have a more anti-inflammatory metabolism by any other reason that are not demographics but maybe diet, that is what is delaying the rebound? I don't know if the question makes sense, but
no, it does make perfect sense, and I do believe that, but there is no data on that. The problem is there is not enough samples of people who stopped antiretroviral therapy to ask this question. What I can tell you is definitely the metabolite we're seeing that is related to time to viral rebound, not only in our published study, because as you can imagine, we did several other studies after we published it, are all really pointing to the gut and gut microbiome. And we know that the gut microbiome is influenced by so many other factors, including the diet, including sexual orientation, including the geographic where you are. But again, there is not enough sample size in analytical treatment interruption study to ask to dissect it that much. In our study, we tried it to, correct for age, gender, ethnicity, art duration, art initiation, your usual suspect. But I really believe that is those other demographic, including diet, including where you are in geography, maybe socioeconomic status, all of this stuff is likely contribute and likely causing a lot of these variability we are seeing. But, we are trying to analyze as many samples as we can get our hands on from different analytical treatment interruption studies done here in the US or internationally, but we are not anywhere close to come to the sample size that would allow us to answer this question. But my thinking without any proof, I believe this definitely has to have a rule, because that is a main mediator of the microbiome.
Yeah, I think it's interesting with L-glutamate, right? It's a new drug now for sickle cell disease. And so apparently, I think it reduces inflammation. It might reduce inflammation because people have less crisis, and I think they kind of fare better on L-glutamate. So
glutamic, as I mentioned, glutamic acid is converted to alpha-ketoglutarate acid, and alpha-ketoglutarate acid usually inhibit NF-κB activation. Now, obviously, in CD4 T cell, that is where result in reduction in viral reactivation. In myeloid cell, that is where result in lower inflammation. So that does make sense, yeah.
Maybe it's kind of interesting, maybe cohort for you to look at some inflammatory factors.
Yeah, I will be definitely interested in that.
Okay, who is taking over from me?
Thank you very much. I'm taking over from you.
Okay, great. Sure. Thank you.
So thank you very much. So just for the sake of time, keeping the agenda, the next speaker is Dr. Lee Campbell. He is from Georgetown University. He finished her BS at Texas Tech University in 2006. And after that, he moved to Georgetown University, where he completed the Master in Science in 2009. And after the PhD degree in pharmacology at Georgetown University. And he did a postdoctoral until 2020 at the National Institute in Drug Abuse and Intramural Research Program. And we were very lucky that he decided to go back to Georgetown, his home institution, and he started as a research assistant professor at the Department of Neuroscience. Lee is very interested in using novel technologies to prove the role of opioids and how this substance interact with HIV in the central nervous system. And he uses very innovative techniques like gene editing strategies, bioluminescence sensors, and genetically modified cell lines. Lee has obtained a DCCFAR pilot award in 2022, focusing in identifying the effects of the contemporary opioids on HIV proviral transcription and neuronal functionality. And thank you very much for being here. We are looking forward for your talk, Lee.
Thanks so much for the introduction, Martha. All right, I'm sharing my screen really quickly. All right, can everyone see this?
Yes, you're
Okay.
fine.
All right, so thank you again, once again, for introducing me. And so the data I'll present today would be based on the DC CFAR Pilot Award that I was awarded in January. So this is really nice because it's sort of like a progress report or update on the research that I've done using the award. So as Marta said, my award or my research project for the DCCFAR was to identify the effects of contemporary opioids on HIV previral transcription and neuronal functionality. So what does it sort of mean when I say contemporary opioids? So if you've been listening to the news, maybe many of you have heard that there's an opioid epidemic, and maybe a lot of you have heard that a particular substance called fentanyl has been causing a lot of overdoses and deaths. So this is well established and put here in this timeline within this review, showing the cases of overdoses over the course of almost 20 years. So starting from the turn of the century, we see the overdoses coming from natural or semi-synthetic opioids. So this probably has to do with the prescription opioids that were prescribed to individuals and then who then abused them. Starting around 2011, we see a rise in heroin. So with more regulations to prescription opioids, people have probably started turning to illicit substances. So that's why we see a rise in heroin. And then finally, starting around 2013 on towards today, we see the rise in prevalence of fentanyl. So what exactly is fentanyl? So fentanyl is a synthetic opioid. And what's special about it is that it has a 100 to 50% higher potency than morphine and heroin itself. And so because of this, and because it's semi-synthetic, a lot of individuals have been using it to lace other substances, such as other opioids or other compounds, including cocaine, methamphetamines, with fentanyl. And because of this high potency and people not knowing they're getting fentanyl, this leads to this increase in opioid-related deaths by fentanyl. All right, so fentanyl, of course, is an opioid, and opioids are often commonly associated with people who inject drugs, or PWID. And so in terms of the health concerns with people who inject drugs, you can look at the sort of health concerns with the drug use itself. So people who inject drugs often go through these cycles of addiction, abuse, and withdrawal, and then sort of like recurring cycles of these three facets. So over time, these individuals may also result in sort of a cognitive decline through the use and this sort of like use and cycling of these different types of drugs. So also with people who inject drugs, there's also a susceptibility to bloodborne pathogens. So we heard, so at least I learned through the DC Drug User Health Scientific Institute group that hepatitis C is a big concern within people who inject drugs, especially opioids. And then most traditionally, with people who inject drugs, the transmission of human immunodeficiency virus is also a big concern. This is primarily spread in this population through the sharing of needles. So a little bit about the human immunodeficiency virus. So once again, it's a virus that infects immune cells, such as the CD4 positive T cell. It kills immune cells over time, and this results in AIDS, or the acquired immunodeficiency syndrome, if left untreated. So once again, AIDS is characterized by susceptibility to opportunistic infection and ultimately death. So a particular part of HIV infection that I study is how HIV infects the brain. So you may not know that the HIV, once after primary infection with the body, can transverse the blood-brain barrier and take residence into the brain very early after infection. There are some reports of it being within 2 to 3 days after primary infection. So within the brain, the virus does not infect neurons. However, these neurons still die over time, and this results in this disorder called HAND, or HIV-associated neurocognitive disorders. So what exactly is HAND? So HAND is deemed a spectrum disorder. There's not just one sort of disability associated with it. Individuals who have HAND can have deficiencies in cognition, including the decreased concentration, forgetfulness, and mental slowing. They can have motor disorders, unsteadiness of gait, clumsiness, and tremors, and also behavioral changes, including switches between apathy, mania, and depression. So what's interesting about HAND and these disorders is that even in the era of combined antiretroviral therapy, these disabilities still persist. So there are still individuals, even though they have controlled viral loads within the periphery, they can still manifest these type of neurocognitive deficits. So now that we know that this is still a persistent disease within the community, how exactly is HIV causing brain damage? So for the sake of this talk, I'm going to focus on one type of cell within the central nervous system that plays a large role in this type of mechanism. And that cell is called the microglia. So what you're seeing here is immunocytochemistry for IBA1, which is a marker for microglia. And so microglia are the resident macrophages of the brain, and they play a role in the immunogenic response within the CNS. So if there's any like pathogen or insult or injury within the brain or central nervous system, the microglia get activated, go to that site of injury and try to clear it up. So what's special about microglia and HIV is that these cells are the cells that can be productively infected with the virus, meaning that HIV can go interact with the microglia, infect it, integrate its genomic material, and then that microglia can produce more virons to further propagate the infection within the central nervous system. So once we know this, that microglia are productively infected, how is that affecting neurons and causing them to die? So over time, it's been postulated that there have been two different mechanisms causing neuronal cell loss by HIV within the brain. One is through the direct mechanism. So this is when these infected cells are then releasing HIV viral proteins or virus itself. And these viral proteins, such as TAT, REV, NEF, and GP120, can directly act on the neuron itself, causing neuronal cell death. In addition, it's been postulated that having HIV within the brain and infected cells causes a constitutively sort of like pro-inflammatory state. And so this is marked by constitutive release of pro-inflammatory factors, including interleukin-1 beta, TNF-alpha, and reactive oxygen species. So there isn't one specific mechanism that that both of these mechanisms work in concert to end up causing synapse simplification and neuronal apoptosis. All right, so now that we know a little bit about drug abuse and how that connects to HIV, the idea is how does drug abuse and HIV are affected in the sort of era of fentanyl. So we know that substance use disorder is sort of like a long-term disorder to manage. So there are a lot of therapies such as opioid replacement therapy or counseling to try to manage substance use disorder. Additionally, with HIV, there's no cure strategy that is widely reproducible and widely accessible to most individuals. And therefore, both of these illnesses can be sort of considered as a common comorbidity. So they're both happening at the same time, basically. So researchers and other scientists, and myself included, had this question: how do opioids affect hand pathogenesis? So in sort of like creating this proposal and being an assistant research professor, the idea is to sort of like come up with your own ideas. And so the ideas and how you come up with them is to sort of identify what's called gaps in knowledge. And so this was something I learned during the grant writing course, and it was produced by the DCCFAR, and it's like a great idea. So the idea is to sort of look at what's been done. So what we're seeing here is sort of like the mechanisms of HIV that I previously described, and what are the sort of papers and knowledge that's already known. So if we look at these 4 different papers that are pretty recent, 2020, 2017, we see certain keywords such as morphine, upregulation or synergism, and we see the word TAT. So that is sort of saying that, number one, a lot of people focus on morphine, which is good because it's sort of the canonical opioid, but not a lot of people are focusing on, these other things that are other opioids that are being seen within the substance abusing population, such as fentanyl. Number 2, a lot of people are focusing on HIV viral proteins or inflammation. So we're down here in the sort of level of mechanism. And finally, that they're sort of causing a synergistic effect or causing more damage. Basically, that opioids are bad. So what I want to do is to identify gaps in knowledge, and what I want to do is to focus on number 1, identifying where these HIV proteins come from, and that was to look at HIV-infected microglia. Can opioids affect the transcription of integrated HIV proviruses, thus creating more HIV viral proteins or creating more of an inflammatory effect within the central nervous system? And secondly, a lot of individuals or published work has looked at neurons and the endpoint, neuronal cell death or neuronal apoptosis. What I want to look at was something before that. How do the neurons function under the presence of HIV-infected microglia and opioids? And so for that, this is what I came up with the two specific aims. Specific aim 1 was to examine the effects of opioids and HIV transcription in a particular cell type called the microglia. And specific aim 2 was to determine the effects of opioids in an in vitro model of neuronal dysfunction. So the main idea was, because of the previous data saying that opioids were bad, we hypothesized that these opioids will increase HIV profile transcription and promote neuronal dysfunction. All right. So going on to specific aim 1. So I need to, so to perform this aim, what we need is something very specific. We need to have a model of HIV transcription in microglia. And so during my postdoctoral fellowship, I sort of made that, and it's called HIV Nanologue CHME5. So the CHME5 cell line, I know, has been sort of controversial, but I think people now have come to terms that it's a rat microglia-like cell line. And so what I did was take that rat microglia-like cell line, and then I integrated a modified HIV provirus in it. And so by doing that, we can have this sort of cell line, a renewable, reusable cell line to test HIV proviral transcription, basically. So how this cell line works, the cell line was integrated with this modified provirus. And so we have the HIV long-terminal repeat, which is the viral promoter. What we took out was the GAG pole, make it non-replicative, but instead we put in nanoluciferase. So nanoluciferase is an enzyme that once it interacts with a certain substrate called cholinterazine, it releases light. Therefore, it's sort of like a very quick and fast way to measure if there's HIV viral transcription. So along with this, we kept the rest of the provirus intact, which contains TAT, Rev, GP120, and NEF, which are all these sort of viral proteins that are associated with neuronal loss in HIV. All right, so how this virus works. So what I did here was to simulate it with lipopolysaccharide, which downstream will act on NF-kappa B and interact with the HIV long-terminal repeat. And what we can see here is that there's an increase in luminescence values after repeated treatments of lipopolysaccharide. So this is suggesting that the LTR is active. We want to also test not just the nanoluciferase, but also the HIV viral proteins that are being produced. And so the one we look at most and that we like the most is the HIV viral protein NEF. So this is NEF staining after lipopolyside LPS stimulation. And here's immunocytochemistry using a NEF-specific antibody for HIV Nanologue CHME5 cells. And so to further confirm that this was actually NEF staining, we did a 50-50 mix with parental CHME5 cells, which don't have HIV in it, and also the HIV Nanologue CHME5 cells. And so what we can see is sort of like this mosaic pattern, suggesting that only the cells with the provirus in it can produce NEF. And finally, the last thing that was of interest to us was how many integrants do we have in the genome of these cells. And so what we're looking at here is by droplet digital PCR, the copies per genome of this HIV provirus that's on the screen. And so what we're looking at is showing that the HIV Nanologue CHME5 has 14 integrants of this provirus, and these number of integrants are stable. So P stands for passage number, so that when you're doing cell culture, you use passagism. Passage cells is basically a way to tell how old cells are. And so from P5 all the way to P25, we see that the copy number of this provirus stays stable at 14 within these cells. All right, so now that I have a model to test aim 1, these are the three sort of experiments I'd like to show to you today. So the first one is, are these cells expressing opioid receptors? So this is a very common sense question, and it was also brought up by the reviewer critiques. And I'm glad that we were able to sort of identify and confirm that they do. So the next one, of course, was the big idea. Do opioids affect HIV transcription in these cells? And then finally, because of the second, the results from the second one, we want to confirm opioid receptor activation. All right, so the first thing we did was to confirm that these cells have opioid receptor expression. And so there were two different assays. One was a Western blot using 15 microgram of protein per lane, and the other one was using immunocytochemistry. And so the primary antibodies for Western blots are here. If you ever need them, the catalog numbers are here. I know that, at least in our experience, finding a good opioid antibody is pretty tricky. All right, so this next piece of data was produced by Uma within our lab, and she performed some Western blots using very interesting controls. So if we go over to the mu opioid receptor Western blot, we see the first two lanes are loaded with cell lysates from HIV nanologic CHMA5 cells. The really interesting control I thought she did was she loaded cell media. So this is the media that cells were actually grown in, so there shouldn't be any type of cells in here. And then as a positive control, she loaded in the last two lanes the mouse striatum. And what we can see here with the mu opioid receptor should be showing around 70 kilodaltons. We see pretty pronounced bands within the HIV nanologic CHMA5 cells, nothing within the cell media, and then sort of these light bands within the mouse striatum. So this was also reproduced with a kappa opioid receptor with a molecular weight of 43 kilodaltons. There's a 39 band, so it should be above this. And we see bands within 43 kilodaltons within the HIV nanologic CHMA5 cells, nothing within cell media, and then we see that again with the mouse striatum. So the delta opioid receptor was, of course, from this the best looking blot. We see the delta opioid receptor should be around 40 kilodaltons. We see it once again above the 39 kilodalton band, nothing in the cell media, and then a little bit within the mouse striatum. So this gives us our first evidence or clue that yes, these cells might express opioid receptors. So the second thing we wanted to do was to determine expression by immunocytochemistry. So this is sort of like the control experiment showing the control experiment for the immunocytochemistry, basically. So cells were either untreated or treated with no primary, so no delta opioid receptor primary, no secondary, so no elixiflor secondary. But if you combine the two, we can see once again that there is immunofluorescence. And so this is what they look like, looking at the mu opioid receptor green merged with DAPI, kappa opioid receptor in the middle, and then delta opioid receptor at the far right. So at least in two different assays, it suggests that there are opioid receptors on these cells, which makes the next part doable, basically. So the next thing we did was to determine opioid receptor based transcription or HIV based transcription in these cells by opioids. And so what we did was run dose response curves ranging from a very small dose all the way of 0.1 nanogram all the way to 100,000 nanogram, which is, if you change that into microgram, it's like 100 microgram per mil as the highest concentration. So the compounds tested were dissolved in water to 1 milligram per mil stock, and then the serial dilutions were prepared. And so in terms of what we tested lipopolysaccharide, which is our positive control. We know this can induce the transcription of the provirus into cells. And then we tested a lot of other different compounds. So of course we had fentanyl here. And so fentanyl, whose therapeutic range is from 1 to 10 nanogram per mil. We chose methadone, which is important for me because methadone is used as a compound in opioid replacement therapy. And so the therapeutic range for this is from 50 to 500 nanogram. Then we used 3 different other opioids that are very specific, for opioid receptors. So Damgo itself is a specific mu opioid receptor agonist. Then we have nalfurafine, which is kappa opioid receptor agonist, and then DPDPE, which is a peptide, is a delta opioid receptor agonist. And so these compounds were placed on the cells for 24 hours. And how these cells were run is because the cells have nanoluciferase that can be plated in a 96-well microplate and then run in a luciferase assay. So this is an example of the cell plating. We have lipopolysaccharide, fentanyl, methadone, Damgo, nalfurafine, and DPDPE in its different concentrations and in duplicates per well. So we ran 2 different plates each time we did it. So one was with just the drugs alone, and then the other one were with the drugs in the presence of 100 nanogram per mil LPS. So this is just saying if there's like this sort of like basal amount of inflammation in the culture, will opioids sort of synergize with this, right? And at the end of the day, we had 3 different cell platings, so 3 biological replicates, and as you can see, 2 different experimental replicates per plate, right? So I wish the results were more interesting, but what we could see is that, number 1, our positive control worked. So lipopolysaccharide had a dose-dependent increase in HIV proviral transcription, and now we're looking at the drugs itself. So even though these cells appear to have opioid receptors, we're not seeing any sort of like significant, or at least seeing any large increases in HIV proviral activity. So if we're looking at fentanyl, or fentanyl with LPS, there's almost basically a straight line. Methadone was a little bit interesting at the very end. So methadone in the presence of LPS did have a 1.5-fold increase in proviral activity. Dango looked the same with or without LPS. Nalfurafine was also the other sort of interesting one that showed increases only within the highest concentration, both within and without LPS. And then finally, DPDPE had sort of like, it seemed to be a separation between LPS stimulated and non-LPS stimulated, but we still need to go back and do a couple more experiments with that. So the idea of this was that, okay, we our positive control is showing that, yes, the provirus is within the cells, it can be activated by something, but with a lot of these opioids, it doesn't seem like they do very much in terms of activating integrated HIV proviruses. So because of that, what we want to make sure was that, were these drugs actually having an effect on the cells? So that was confirming the receptor activation. So this is ongoing work right now, but we have a very small experiment where we performed a time course looking at receptor localization. So opioid receptors are G protein coupled receptors. And so if they are acted upon a ligand, it suggests that there's 2 different sort of signaling pathways. One is through the G protein, and then another that can be seen is through the beta-arrestin internalization pathway. So the idea was we would treat these cells with compounds such as fentanyl or methadone and see the localization of the receptor. Are they changing? All right, so we did that experiment, and I'll show you methadone first because it's the most pronounced. So this is the time course looking at 0 minutes, 5 minutes, 10 minutes, 15, and 30. And what I hope you can appreciate is that the localization of the opioid receptors starts to get a bit different. They seem to be more spread out in a very apparent sort of like circle around sort of like the nucleus, and then we start seeing it become fragmented after 10 minutes, 15 minutes, and it's very apparent at 30 minutes. So this is suggesting that, yes, methadone is acting on opioid receptors and doing something. The same thing can be seen with fentanyl, except it happens a little bit faster. This sort of fragmentation or difference within immunofluorescence is seen as early as 5 minutes and proceeds from 10 minutes and 15 minutes. But oddly, we didn't see as many cells with this pronounced effect by 30 minutes. Right. So at the end of the day, what I like to show from these experiments is that, yes, these cells do have opioid receptors, which is confirmed by Western blot and immunocytochemistry. For whatever reason, the opioid receptors have a minimal effect on HIV transcription. And at the end of the day, the opioid receptors are having an effect on cells, as shown by time-dependent changes in receptor internalization. So for the future experiments, of course, if I'm saying that, okay, opioids don't really have an effect on proviral transcription, I want to show more in terms of intercellular mechanisms. We know that the HIV long-term repeat has specific binding sites for relevant transcription factors, such as AP1 and kappa B. So if we can show that receptors are being activated, however, there's no effect on sort of like these canonical transcription factors, that might be a reason why we're not seeing any type of opioid effects within these cells. Finally, once again, with the rigor and reproducibility, this is a cell line. I'd like to sort of change gears and try to recreate the big effects that that we're seeing within our data to a more relevant model using primary microglial cells. All right, how much time do I have left? Should I stop here? I don't want to get out of time.
Yeah, I think it's time probably to stop. Yeah.
Yeah. Okay. I'll stop here. So I had other things, but I will go to the acknowledgements. All right. So yeah. So this has been a really great learning experience since I'm, of course, a junior faculty. This is the first time I was able to, have an idea that I think might work, and then do everything that is associated with that. From having a budget and being able to order the compounds, to saying, okay, these are the experiments that are working, these are not. Like, how do I pivot and make it a useful piece of data? And from that, I'd like to once again thank my mentor, Dr. Idla Muketti, the people within our lab, Andrew Spidell, Chrissy, Melanie, Sophie, and Uma, Dr. Adam Cacavano. Hopefully, in future experiment or future iterations of this, I could show the second aim, which is, I think, pretty cool. Of course, the DCC-FAR for all their mentoring thus far within this process, and of course, the DCC-FAR Pilot Award. All right, thank you.
Thank you very much. So anybody has a question for Lee? Okay, Manya,
Hi.
go ahead. Thank you.
Thanks so much for this terrific talk. I love to see the intersection of our clinical world with the basic science world. So this was really fascinating. We're currently doing a lot of studies, particularly in the field, even in DC, with people who inject drugs population and with and at risk for HIV. So this is really close to home. I might have missed it, and I'm not a basic scientist, so please forgive me if I did. But I'm interested if you were able to look at the relationship of more than just one drug at a time. So many of our participants don't just use fentanyl, they use fentanyl and something else. The combination polypharmacy is a real issue. And the other thing is, I may have missed it also, but I'm curious if you looked at buprenorphine in addition to methadone.
Sure. So question number one, yes, combining different types of drugs for polydrug abuse, especially in this assay, since it's just cells on a plate, you could put whatever combinations of drugs. And if you have an idea of saying, okay, in the population, we're seeing these two drugs being put together, yeah, I'll go ahead and just put those drugs together in a dish and see what happens to profile activity. And so number two, in terms of buprenorphine, that's also like a learning thing for me. So if I want to get buprenorphine, I need a Schedule 3. So we have schedules up to Schedule 2N for drugs. So when you're ordering drugs, you can't just like say, hey, I want illicit substances from Sigma, right? So you need to have DEA approval. And so we could get all those drugs that were listed and oxycodone, but we couldn't get buprenorphine because that's Schedule 3. And so I have to work with the DEA to get licensing for that drug.
Great. I'll reach out to you because this might be a really great area of collaboration. But it sounds like you're working with colleagues in the drug user health group, but this could be really interesting. And we do have ideas. We have data on what type of polypharmacy people are using, which are most common, and that might be really important. But thank you very much.
All right, thank you.
Thank you. We have another question. Silas, I hope I pronounce well your name. No? Do you have it?
Yeah, it's Selassie. Thank you.
That's it. Sorry.
Thank you for the presentation. I have a question and then a suggestion. I realized that you saw well, you didn't show beta actin or or any housekeeping gene, but I realized that just from looking at the bloods, you may have more receptor in the model cell than in your positive control. do the model cells naturally express such a high amount of those receptors?
Sure. Okay. So to answer that question, I think it also has to do with the, I guess what we're talking about, the positive control, which is the mouse striatum, right? So
Yes.
when we're loading HIV nanoleuks C5 cells, it's like one cell type. And so I think through other data, I've shown that they have, opioid receptors. So that's just sort of saying like it's concentrating one type of cell for the assay. When you're loading in a mouse striatum that contains microglia, astrocytes, neurons, and depending on the area, maybe high or low amounts of opioid receptors. So
Okay.
that might be a difference between the relative levels. Why it may seem like, oh, these cells have a lot of opioid receptors when it's not a one-to-one sort of like comparison. So if I said from the striatum, I dissociated the cells and only took the microglia out and then compared them, then that might be a bit more of a one-to-one. But right now, just saying a homogenized mouse striatum has, once again, microglia, astrocytes, all the different types of cell types.
Okay, I totally understand. And secondly, to my question stroke suggestion, from your time course experiment, it seems that the receptors get activated in a very short time, I say maximum 30 minutes. And even with the case of fentanyl, you seem to lose the activation after 10 or so minutes. So maybe it may be good to not to stimulate the cells for 24 hours, but to try the activation for a shorter time course. Maybe that may, I don't know, you may see the same results or you may see different results. That just occurred to me.
Yeah, no, that's great. Yeah. I think the original reason why we chose 24 just starting off, because that's sort of like historic of what I did with lipopolysaccharide. So I treated with lipopolysaccharide 24 hours later, I could measure, the HIV profile response and that it was up. And so that's, I guess, why we stuck with that. But what you're saying is comparing or combining the effect that we see with our time course, our short time course, seeing that the receptors are already doing things, right? And then also looking at proviral activity at that time point would, definitely be useful.
And so finally,
well, as Celeste, sorry, I'm going to stop you because of time.
Sorry. Yes.
Sorry.
All right, we'll have to move on to the next round.
To move to the next because we are short on time. But I transfer to Alberto now. But thank you very much, Lee. If anybody has additional questions, you can put it in the chat.
Okay.
Yeah, thank you. Sorry about that. It was really interesting. I actually have questions for you, Lee, but I hold them and I email you later. So now we are going to pass to have two short talks by two of the two new T32s that are both one at Georgetown and the other one at George Washington University. So the Georgetown has a T32 for graduate students. This is a training program in neuro-HIV. And the first speaker will be from that program. So let me introduce Griffin Greco. Griffin did a Bachelor of Science with specialty in neurobiology at Georgetown University from 2012 to 2016. After that, he was a research assistant at Walter Reed Army Institute of Research. And in 2018, he joined back Georgetown University to do his PhD. And right now he's a second year in the training program in neuro-HIV. And the title of his talk is The Role of CCR5 in Promoting Neuroinflammation in an ApoE mouse model of Alzheimer's disease and its relevance to HIV-associated neurological disease, or HAN. And with that, please, Griffin, if you can start your presentation.
Thanks so much. Yeah, I'll share my screen. All right, can everyone see that okay?
Yes.
Okay, great. Yeah, so thank you very much for having me talk. The title of the presentation I'm going to present today is Investigating the Role of CCR5 in a Mouse Model of Alzheimer's Disease. So just a little quick brief background on Alzheimer's. It's the leading cause of dementia, characterized by learning and memory deficits. The hallmarks are extracellular amyloid beta plaques, intracellular hyperphosphorylated tau, neuroinflammation, and cortical and hippocampal atrophy. And this is just a graph of stratified age groups projected to be diagnosed with Alzheimer's through the year 2050, when it's going to hit roughly 14 million. So it's a very prevalent issue, and that's why I've decided to study that. So ApoE is the single most important genetic locus for determining whether someone will develop Alzheimer's. And just a single ApoE4 allele increases your risk by 3 times, and a homozygous ApoE4 carrier has a 12-fold increased risk. And this is the graph showing the percentage free from Alzheimer's as you age. And as you can hopefully see, the 4-4 genotype has almost no chance of not developing Alzheimer's as they age past about like 80 years old. In the brain, ApoE is important for binding low and high density lipoproteins and clearing them from the brain. And it's thought that maybe having the ApoE4 allele may impair the clearance of A beta from the CNS, because amyloid can also be detected earlier in E4 carriers compared to those with E2 or E3 alleles. I'm sorry, I'm moving my cat. So one deficit that ApoE4 mice have is in gamma oscillations. And gamma oscillations are basically activations of excitatory inhibitory neurons that oscillate between 20 to 80 hertz. You can subcategorize it kind of as low gamma or high gamma. And these gamma oscillations are very important for cognitive processes, including working memory. And this was an excellent paper by Emily Jones, who did multi-electrode implantation and showed that in the CA1 region, CA3 region, and in the dentate gyrus, ApoE4 knock-in mice had significantly decreased slow gamma power between the ages of 12 to 18 months, and that these gamma deficits earlier in age could actually predict later cognitive deficits in those same mice. So one of the important things that's relevant to generating gamma oscillations are perineural nets. So perineural nets are composed of several different proteoglycans bound to a hyaluronan backbone, and they're localized to the soma and proximal dendrites of fast-spiking inhibitory interneurons, such as parvalbumin interneurons, that are critical for generating these gamma oscillations. And I'm not going to go into a ton of the details on this paper, but essentially they used an enzyme, chondroitinase ABC, which will enzymatically digest the backbone and degrade those perineural nets. And when they did this in a model of monocular deprivation, which is supposed to stimulate a relative period of neuronal plasticity, when they administered this chondroitinase ABC, they saw a significant increase in the amount of gamma power that they were able to see, indicating that they had a more increased potential for plasticity. So one of the things that can affect perineural nets and is a very heavy focus of our lab and the Kathy Conant lab is matrix metalloproteinase 9 or MMP9, which is produced by neurons in an activity-dependent manner. And like I said, it's important for the cleavage of chondroitin sulfate backbone of perineural nets. And it's secreted as a proenzyme and then activated extracellularly. But its activity as an active enzyme can be modulated via inhibition by tissue inhibitor of metalloproteinases, or TIMPs. And so this is the focus of what I have been looking at. So this is some previous work by a graduate student who's now graduated from our lab, who was looking at this in a corticosterone model of depression. And she saw that using venlafaxine, which she also showed increased MMP9, was able to significantly increase the PV to PNN ratio, which essentially means that she saw less perineural net intensity staining in this. And it's not shown in these graphs, but she did show that this effect was MMP9 dependent. She did that using an MMP9 antagonist. And so then tying this back to my project, looking at the role of CCR5 in all this. So CCR5 is a chemokine receptor expressed on microglia, astrocytes, and neurons. It's necessary for the HIV infection of microglia in the brain. And recently it's been shown to be an important regulator of neuronal plasticity, and I'll get into this a little bit more soon. And CCR5 antagonism is also shown to decrease extracellular matrix deposition in liver and kidney. So that's thought to be why this could potentially be interacting with perineural nets in the brain. So yeah, just briefly, this group showed that knocking down CCR5 increased mice in contextual fear conditioning, as well as Morris water maze and a social memory task. So knocking down CCR5 significantly improved all of these cognitive deficits. And this was the paper that got me interested in it because it was talking about the role in post-stroke recovery. And they found that, so in this, they used both a short hairpin RNA knockdown of CCR5, and they used Maraviroc, which is the FDA-approved antagonist for CCR5, often given to treat HIV. And so they showed that with both of these knockdowns, they had significantly better behavioral improvements. And this came about from the finding that actually in humans, clinically, people who had a Delta 32 mutation of CCR5 had better post-stroke recovery. So that's what led them to investigate this. And that's what led me to my interest in this in Alzheimer's. So we actually saw that, so on the left is preliminary data we collected from astrocytes, either expressing the human ApoE2, E3, or E4 allele, and we saw that they had the E4s had a significantly increased amount of CCL5, which is the endogenous ligand for CCR5. And another paper also found this as well in astrocyte lysates as well. So that leads me to the aim I'm presenting here, which is to determine whether CCR5 antagonism in ApoE4 targeted replacement mice affects these molecular targets important for cellular plasticity. So the methods I'm presenting first are from CSF samples collected from Alzheimer's patients. So they were enrolled in a clinical trial for resveratrol, but that's not relevant to mine. All the samples that I used were from baseline prior to either placebo or resveratrol. And I utilized these to do ELISA to look at the CCL5 levels, TIMP1 levels, and TIMP3 levels, zymography to look at MMP activity, and Western blots for perineural net proteins. So for the ELISA results, so firstly, you can see that RONTES is significantly increased in the E4 compared to both of the other E3 and E4 genotypes. TIMP1 is also significantly increased, and TIMP3 is also significantly increased. And then we didn't see a change in MMP1, and I'm going to go into MMP9 in a second. But this is important because essentially, if we're seeing significantly increased levels of TIMP1, then that means that there's significantly more inhibition of MMP9 if we don't see a change in that. So to look at those, we did zymography and Western blots. So zymography, basically, you load the CSF, and it's a digestible gel. So when you see more of the band being digested, that indicates that there's more active protein in that. So we didn't see significance, but we did see that there was a trend towards a decrease in the E4 to have less MMP9 activity. And then in the bottom image is a Western blot, a representative Western blot from those CSF samples as well, for Brevican, which is one component of perineural nets. And we did see a significantly decreased amount of the cleavage of that, that's what you would see in the CSF. So indicating that in the 4-4s, you have less cleavage, which means the perineural nets in the brain are likely more rigid and more intense. So now another method that I'm using is EEG recordings to actually look at the gamma power that I was talking about. So this basically involves implanting a wireless device into under the skin, and then we have electrodes placed on the surface of the scalp, and then they're continuously monitored over a long period of time to record their EEG activity, which can then be filtered out for gamma frequencies. So the first two cohorts I did were from just wild type mice compared to CCR5 knockout mice. And I found that over the course of these roughly 2-week recordings, the CCR5 knockouts did have significantly higher gamma power compared to the wild type mice. And the gamma power I measured was a slightly lower range of gamma. It was from 20 to 55 hertz. And so then this led me to create ApoE4 and CCR5 heterozygous mice. So they have a heterozygous knock-in for one ApoE allele and a heterozygous knockout for one allele of CCR5. And then I performed the same EEG recordings on those. And interestingly, I found that the E4 mice that had one allele of the R5 knocked out had significantly higher gamma power than just the E4 crossed with wild type mice. So only one knock-in of the ApoE4 allele. And then in these mice as well, I also looked at TIMP1 levels, and I found that the TIMP1 levels in the E4R5 were also significantly lower than in the E4 wild type heterozygotes, which again is implying that there's higher MMP9 activity in those mice, which would lead to less perineural net intensity. So just a quick summary. So we find Rontes, TIMP1, and TIMP3 all significantly increased in ApoE4 Alzheimer's patients compared to 3.3 and 3.4 patients. The ratio of active to pro MMP9 activities trended towards a decrease as well. And we find that brevican cleavage was significantly decreased. CCR5 knockout mice have significantly higher gamma power than their age-matched wild types. And the E4 CCR5 heterozygote mice also have significantly higher gamma power than their age-matched E4 wild-type heterozygotes. So future directions, I want to administer Maraviroc, similar to that other paper I presented, just to make a more translational component to my research, to examine changes in MMP9, TIMP1, and brevican, all these important molecular markers, as well as analyzing those in the heterozygotes. I would like to analyze the gamma power on another cohort of the E4 heterozygotes. My N was only 3 for those. So although I did achieve significance, I just want to reproduce that. And then I also want to perform behavioral tests with these mice with EEG devices to see if that increased gamma power is actually correlating with better performance on working memory tasks. And so my acknowledgements, my mentor, Dr. Kathy Conan, everyone in my lab, my committee is Dr. Bill Rebeck, Italo Muchetti, and Dr. Scott Turner. These are some other people who really helped me with my project and my funding, the T32. So with that, I'll be happy to take any questions. I hope I didn't go too far over.
No, you're perfect on time. Anybody has any question?
I actually have a question. Well, kind of just honestly about your last slide with what you intend on doing in the future. I'm not familiar with really any of this and any mouse work or anything. So what kind of behavioral test? Is there like a standard like kind of panel that, you know?
Yeah, so similar to like with the behavioral tests that were done in the paper I presented, fear conditioning is one where you can put a mouse in an electrical chamber and they'll get zapped, and then you test them again some period of days later to then see how their latency to freeze is. So that will kind of be a measure of their long-term memory.
Okay.
The Barnes maze is one where you have them search for a reward in a hole in a hole on a table. So it's a measure of spatial memory. You can do that. You can do social memory as well, where you habituate a mouse to another mouse, and then later move that mouse to a new mouse that he hasn't met yet, and see how much they spend time exploring. So there's several different correlates of memory that you can test.
Gotcha. Okay, awesome. Thank you.
Any more questions? Well, if you have any question, you can put it in the chat or directly to him or to the group. And now I'm just going to introduce our last talk from the T32. So our next speaker is Teresa Murphy. Teresa Murphy did her bachelor's in biology at the Pennsylvania State University. where she also worked as an undergraduate researcher in the laboratory of Dr. Robert Paulson. And in 2020, she joined George Washington University and the Institute for Biological Sciences in our PhD program. And she was one of the first recipients of the T32 on HIV persistence, comorbidity, and treatment. And today, she is going to be talking about utilizing broadly neutralizing antibodies for HIV cure strategies.
Okay. I will try to keep this tight since we're running behind. So good afternoon, everyone. As Dr. Bosque said, I am Teresa Murphy, and I am a graduate student in Dr. Rebecca Lynch's lab at GW. And today I'm going to be presenting to you all on my T32 project, which is utilizing broadly neutralizing antibodies for HIV cure strategies. And now just to go into a little bit of background first, today we're going to be focusing on the envelope of HIV, which is encoded by the ONV gene. And the envelope is the target of the immune system, and of interest to us, obviously, is it being a target for antibodies. And the protein is made up of a GP120 trimer and a GP41 transmembrane trunk. And this protein is very diverse due to the error-prone polymerase and the high levels of replication of HIV. And all of these factors combine to form a protein that is very, very good at viral escape from the immune system. And just to underscore the diversity of the envelope, within one individual, the ONV gene can differ by up to 2.5%. And another layer of difficulty for diversity is also global diversity of HIV. And this is broken down into different subtypes and recombinant circulating forms of HIV, in which the ONV genes can differ by up to 30%, really showing that there is a need for a broad treatment if we are going to use antibodies for cure strategies. Another issue with global diversity is that historically, much of the research that has been done has been on subtype B viruses, which, as you can see here, is most prevalent in the Americas and Western Europe, but that is not the case globally. And so much more diversity in research is needed to develop cure strategies. And a new player in the last decade or so on the cure strategy front has been broadly neutralizing antibodies, or BNAbs. And these BNAbs are antibodies that are very rare and isolated from chronically infected individuals living with HIV. And these antibodies have the ability to neutralize greater than 50% of viruses on a predetermined global HIV panel. What is important to note, though, is that these individuals who are generating these BNAbs are not cured of HIV because since their viral quasi-species has been exposed to these BNAbs for long periods of time, they are able to escape. And these BNAbs tend to bind to 5 major conserved epitopes, those being the V3 glycan supersite, the V2 apex, the CD4 binding site, the membrane proximal external region, and the interface between GP120 and GP41. And as I mentioned, they have begun to be used in clinical trials in people living with HIV. And these clinical trials tend to come in 2 major flavors, those being ART interruption, where someone who is virally suppressed is given a broadly neutralizing antibody after being removed from antiretroviral therapies, or in viremic individuals where an individual who is ART naive and has a high viral load is given a broadly neutralizing antibody treatment. And the one thread linking all of these clinical trials, no matter the setup or the broadly neutralizing antibody chosen, is that these treatments are offering transient viral suppression, but viral rebound is almost always occurring due to viral escape. And our research question for this is, how can we take advantage of the fitness costs associated with viral escape from BNAbs for designing combination treatments? And to do this, we have been designing an in vitro viral escape assay in which we incubate a single known virus with a predetermined suboptimal concentration of a chosen BNAb. And after this incubation, the virus is used to infect ex vivo CD4 T cells, and then they are cultured in the presence of the chosen broadly neutralizing antibody. And over time, we are monitoring the replication kinetics of the virus and steadily increasing the antibody concentration to kind of teach the virus how to escape from the BNAb so that we can see what common escape pathways are occurring and if we can use those to our advantage. And we are measuring this viral resistance via genotypic and phenotypic assays. And in the work I'm going to be showing you all today, we used an infectious molecular clone with a subtype B NL43 backbone that had a subtype B YU2 envelope cloned in. And we chose two very well-studied common BNAbs for our use in these experiments, which are 10-1074, a V3 glycan antibody, and VRC01, a CD4 binding site antibody. And so first I'll go over our VRC01 results. And what we did was we started our experiment using 4 different suboptimal inhibitory concentrations or ICs of antibody that were chosen based on previously done pseudovirus neutralization assays. And at the 2-week point, we wanted to choose which concentrations had less replication than wild type, just so that we knew that there was some level of neutralization happening, but the virus was not completely neutralized by the concentration that we chose. And what we noted was that around day 40, we are seeing a large jump in replication in the presence of very high antibody concentrations, leading us to question if this is when we are forming resistance. And what we saw using our TZMBL pseudovirus neutralization assay was that we did see complete resistance forming by day 45 with 0% of the virus being neutralized by VRC01, whereas at day 30, we only saw minimal resistance forming. And when we look at our sequences, what we see is that at day 40, or sorry, I apologize, at day 30, we have a mutation appearing at position 561, which has been previously shown in a paper to give partial resistance to CD4 mimetics. But then by day 45, we have 2 mutations occurring in CD4 contact sites where VRC01 binds closely to, most likely leading to the complete resistance that we saw. And here we just have a summary of the all of the mutations that we saw and their locations. And we had 2 non-antibody specific mutations occurring that occurred in our wild type samples. And we plan to look into those mutations in the future. And now moving into our 10-1074 experiments, we did the exact same methodology setup as I explained in VRC01. And what we saw was that similarly around that day 38, day 40 marker, we are seeing high levels of replication in high antibody concentration. And as seen prior, we are seeing complete neutralization resistance in both of our experimental conditions, whereas at day 30, we saw absolutely no resistance. And then that is backed up here by the fact that at our day 14 and day 30 sequencing time points, we are not seeing any antibody-specific mutations appearing, only this non-specific mutation that occurred also in the wild type samples. But then by day 45, what we are seeing is a mutation occurring at position 334, which is on the N332 glycan, a major mutation site for 10-1074 escape that has been seen prior. And our conclusions from this work is that in these preliminary experiments, we were able to generate mutations in vitro using antibody pressure in our new assay. And we were also able to recapitulate previously seen in vivo mutations in our in vitro assay. And some of our immediate future directions that we plan to do are, first of all, we are repeating our YU2 experiments to determine if we are able to reproduce the mutations that we saw. And then we also plan to study different infectious molecular clone and BNAB combinations to determine common escape pathways. And finally, we plan to utilize donor outgrowth viruses of genetically diverse subtypes to study common escape pathways to design possible combination treatments. And with that, I would just like to thank my lab, especially Dr. Lynch and one of our undergraduates, Jordan Kimner, for all their help in this work that I have been doing. And I would also like to thank the GW HIV Persistence, Comorbidities, and Treatment Training T32 for funding my education. And thank you.
Thank you, Teresa, for being on time. And then anybody has any question? I'll start with the first one. So Teresa, you are only mutating the envelope. Do you think that any other parts of the genome, maybe there is some compensatory mutations that can also explain some of the escape?
Yeah, so we've thought about that. As of right now, we are only focusing on the envelope, but in the future, we may expand to look at different parts of the genome to see if there are other mutations occurring at other parts.
And then all this was done with senior sequencing, right?
Correct, yes.
Like the future is doing the ISeq?
Yes, we are still getting that off the ground. It's surprisingly more difficult to get it off the ground than we thought it would be, but we're working on it, hopefully by September.
Okay, anybody? Any question? Well, if there is no more questions, I will pass the baton to Sergey and I'll let him.
Okay, thank you. Yeah, I think we're coming to the end. Should I show my slides? Yeah, right. Okay. All right. So let's see if I can. How should I do it? One sec. I have to take over, right? Take over. Where's this? Sorry. Reactions. Okay, share screen. Okay. Okay. Share. And share. Okay, so I think that's the end of our program. So basically, I'm going to highlight our basic science core activities and funding which is available. So we have a number of core services available for the DCCFR users. So those which are marked in bold. So these are cores which receive some support from the DCCFR. So these are virus detection lab and analysis at GW with Michael Bukrinsky. There is also next-gen sequencing lab with the capability of single-cell sequencing with Jean Jordan. There is a multiparametric flow cytometry also at GW. I think it used to be with Dr. Bosky, but I think it's moved to someone else now. And then we have also RC Proteomics core at Harvard University. So, so apart from those four services, we have also clinical virology core at Veterans Affairs. There are a number of labs at GW, including pathology core, which can let you do prepare slides, which are actually quite nice. We used it. There are some additional flow cytometry core labs, one at GW, and the other one is, I think, at Georgetown. There's very strong and very extensive microscopy and imaging core at GW. And then at Harvard, we also have this interesting core where you can do the in silico drug design, and you can use machine learning and artificial intelligence to basically screen and design drugs. There is this new, relatively new imaging and then biocore services available at Georgetown University. So, so those cores, they employ a number of instruments. So these are available instrumentations, probably not all, but I think the major instruments. So these are include different fax machines, various sequencing platforms, various imaging platforms, mass spectrometer at Harvard, real-time PCR platforms, and then flow cytometry, as I mentioned, and the platform to study molecular interactions, like these are biocore instruments, and then there's also MRI scanner, and then interesting capability at Georgetown where you can do some, I guess, MRI analysis and then brain scanning analysis. So all this work can be done with the support of the admin core. So basically, you need to apply for a microgrant program. There is a link where you have to go through the DCCFR website. Basically, you just fill out the form, and then you'll be contacted, and then you need to fill out kind of it's a relatively brief application, and then it will be reviewed, and then you most likely will get funded. So you can ask for $2,500 for the regular service, and you can ask for up to $5,000 for more expensive service, like for gene sequencing analysis. So we also have pilot award programs. So these are basically provided to the investigators who are either new or trying to move from another field. That there is another mechanism which I'm going to mention. But so those are reviewed 3 times a year. The pilot awards are $50,000 per award, and we have now the cycle for the full award open. So the pre-submission is September 8th, and then full application will be due in November 9th. And so we also have this Rabbit Awards. So again, this is the very small award, $25,000, but it allows basically for, I guess, very focused question to be answered to basically in relation to the HIV epidemics. Again, the cycle is now open. The event, the letter of intent is due in September, early September, and then full proposal will be due end of September. And I think that's it, right? Okay, so I think with this, I'm going to thank all of you who stay long, and then I think we have very interesting presentations, and I hope those presentations will spark some activities, some new interests, and hopefully we'll collaborate more and write more papers and publish more grants and move forward as CFAR program. Thank you so much. Okay.
Well, thank you, Sergey. Thank you, everybody, for attending, and thank you for, of course, for the to the speakers.
For the presenters, yes. Thank you very much.
Okay. Bye bye. Bye bye.
Bye bye.